the book is divided in 5 parts. There's an introduction section. The first part is dedicated on pharmacodynamic biomarkers, also known as PD biomarkers. Then part 2 is predictive biomarkers, predictive of a drug treatment effect, for example. Part 3 are surrogate biomarkers. Part 4 is how to combine biomarkers, make a composite model or a signature. And part 5 is about writing an analysis plan. And the my background is actually spanned almost 20 years now, between clinical biostatistics, translational statistics, and data science. So the role of biomarkers has actually steadily gone up and become more important and powerful in drug development, impacting clinical biostatistician, biomarker biostatisticians, as well as data scientists working in the clinical trial space. A bit about me. So as I've mentioned, I have been working in the pharmaceutical and molecular diagnostics industry for a while, and been focusing on biomarker, both in the development of drugs as well as tests, high dimensional tests, for example, thyroid cancer, or any algorithms that combine SNPs and so on. Currently, I'm head of statistical methodology and data science in neurology at ASI, where I lead a team where we're focusing on creating artificial intelligence. And by that, I mean machine learning and deep learning methodologies and strategies for looking at the diverse set of structured and unstructured data that are biomarkers that are coming in through our clinical trial. I'm also leading statistical innovation in trial analyses, trial data analyses. I have some background in academia. I completed my postdoc at UC Berkeley in statistics after I completed my PhD in biostatistics at Harvard School of Public Health. I've also taught at UC Berkeley, and one of my favorite courses that I taught there was machine learning. Recently, I've published a book in biomarker analyses in clinical trials using R. It came out in April 20 from Taylor and Francis. So today's webinar is going to be focused on that book. This is a circle or a wheel, rather, that I came up with. And this wheel says that for the statistician working on biomarker analyses, it is a wheel of collaboration and innovation. So we have here the biometrics and data management group in a drug development environment. And the folks working here, which are people like us, I'm assuming most of us here in the audience, and I believe there's, Rick mentioned there's more than 100 people signed up for this course, are data scientists and biostatisticians, bioinformaticians, and so on. And we will be working very closely with translational science when a biomarker or biomarkers are being developed for a clinical program. We will also work with the bioanalytics, the lab services for PK/PD biomarkers, safety biomarkers, and so on, clinical operations in order to, when the data is coming in, and in developing assessment schedules for collecting the biomarkers. And we would work with commercial groups for observing those biomarkers in real world data, the clinical science or clinical development, the physicians, we are always working with them in the development and development of biomarkers in understanding the disease and how the treatment affects the disease process. And finally, finally, I wanted to talk about the regulatory. The regulatory science is very much involved in developing an overall strategy for biomarkers. So especially if it comes to that the biomarker will be in some way involved in the submission for the acceptance of the approval of the drug. Some of the examples here that I have mentioned, and these are just examples, these are not exhaustive list of what kind of work that the statistician is involved in. The whole biomarker-based drug development here just says that all phases of drug development now include biomarkers. It doesn't matter whether it's oncology or cardiology or immunology or neurology, where I'm currently working. The trial design may be influenced by predictive biomarker. So right from the design, we're going to talk about it a lot today. Is it going to be biomarker positive group, biomarker negative group, and so on? Predictive model development is something I'm very much involved with right now, which is combination of biomarkers using machine learning. Is there a signature of a group that's going to get worse very fast in the disease, for example, and it's a disease progression model. And last but not the least, imaging is becoming very much important in clinical trials. The imaging as a modality is being used from PD to surrogate to even predictive biomarker. We won't talk about imaging as an unstructured image today, but I see it also as part of biomarker development for drug development programs. So the whole point of this slide is to really show how much collaboration it takes with different functions. Drug development is a multidisciplinary effort, and it definitely takes collaboration for the statistician with all the functions to drive biomarker development. And innovation in terms of statistical methodology is very important, because as there are more and more biomarkers, the methodology also has to improve and keep up with the technology. So what is a biomarker? And the FDA defines it as a characteristic that is measurable. So it's a quantifiable, measurable, objective evidence of a biological process or a disease process or response to a drug. It can be molecular, histologic, radiographic, physiologic, and however, it's not an assessment how the individual or the subject or the patient feels, functions, or survives. So it's more of an objective assessment, measurement, not a subjective one, like the patient-reported outcomes or PROs. So lot of folks understand biomarkers to be mostly genetic biomarkers, and I've included them here, mostly from the oncology field, where biomarkers have been heavily developed and used. And even within genetic biomarkers, there are many different types of biomarkers. They could be tissue histologic, they could be blood biomarkers, any fluid. For example, the cerebrospinal fluid, which is often we have CSF-based biomarkers and blood-based biomarkers in Alzheimer's disease, for example. But even without and outside of genetic biomarkers, we can have lab-based biomarkers, right? Lipids is an example of that. Smoking status could be a biomarker. Some of the newer modalities include, as I mentioned, radiographic, like PET and MRI images, or it could be captured by a single numeric measure, like overall plaque burden in Alzheimer's disease. It's derived from the PET image. So that's a radiographic biomarker. Composite biomarkers is a combination of biomarkers, and now, and also this new modality called digital biomarkers. And the digital biomarkers is not something I will talk too much about today, but it's becoming more and more prevalent. And how it's defined is it's any information being collected by wearable, portable, injectable, or digestible devices. So any information coming from digital devices worn or digested or implanted or worn by the patient. So you can see it's a broad collection of broad range of sources of information right now that we have available, that we call biomarker, for that is available from the subject to study. Some of the fundamental pharmacology, let's talk about how it starts, preclinical and also in the early human trials. We're interested in the pharmacological questions that have to be answered. These were some of the original use of biomarkers, right? Drug exposure, for example. So drug exposure at target, and so is the basically the target, and then target engagement, target occupancy, how long and how much of a target protein must be engaged by the drug. And then the last bullet is PD effect. So PD is a pharmacodynamic effect of being treated by the drug. It could be a proximal or distal pathway of the target. So basically, the PD biomarkers is biomarkers that measure the pathway downstream of the target protein. These are some of the fundamental questions. So what I talk about in the book is the evidence of all the evidence must be looked at together to make a decision about the drug and or the disease. So it's an integrated framework of looking at biomarker. Integrated meaning across the different phases. As we get more and more data, we have more information. Also, there's number of biomarkers, and each biomarker will provide some information about either the disease or the drug. So several PD biomarkers, now it's very common to have several pharmacodynamic biomarkers over several phases of clinical development, and it constitute a complete picture of the drug's mechanism. So now that the science is progressing, people are interested in and actually do understand more about the disease. They want to know more about the disease, and also they want to know how the drug or the pathway that the drug targets interact with the disease. So we want to know the drug's mechanism in the body from minutes to hours, days of drug administration. So we could be, for example, the drug development may be concerned with validating the target engagement. We will be called in to assist in selection of the optimal dose, depending on how the target is being modulated. And we have to identify early markers for early efficacy or safety. We could be involved in identifying a patient subgroup who is responding to the drug or who is having a safety event and should be removed from the trial or monitored. And also biomarkers will be are studied to see if a combination of drugs are going to be suitable for treating the patient. I just want to quickly mention that there are 3 pillars that Pfizer has put out, and I've discussed this in the book, that there are 3 pillars of survival of a drug through clinical development in order to progress to phase 3. We all know very well, we are in the pharma industry. Many of us are, I'm assuming most of you are. So the drug must demonstrate through these different phases that there's enough exposure at the target site of action over a desired period of time. So certain analyses have to be done to that data to make that decision. Then it has to bind to the pharmacological target as expected for its mode of action. And then it has to express itself, the activity, commensurate with the demonstrated target exposure and target binding. And these goals are achieved, as I had mentioned, by making assays or contracting with vendors who have validated assays for certain pathways and targets, certain biomarkers, and measuring them in clinical trials. Okay, with that introduction, one of the reasons I wrote the book is to really summarize some of the analysis that is routinely done, and I've chosen R to exemplify those analyses. So let's start with the pharmacodynamic biomarkers. So there's an ecosystem of tools predicting risk and progression. Okay, so the outline, the first outline of this subject, of this part, is that it could range, the PD biomarker could range from proof of mechanism to proof of efficacy. The same biomarker could be proof of mechanism or proof of efficacy, but usually not. There are multiples. There's usually one, maybe two proof of mechanism and many proof of efficacy biomarkers. We may be called in to start and help studying them in animal toxicology studies. We may be called to design bioequivalence studies. We have to help visualize the biomarkers over time. And one of the things that I'm very interested in is joint modeling of the biomarker and the efficacy endpoint. So rodent toxicology studies are usually precedent. So they take place before the clinical PK studies in human. And drug exposure measured by concentration, as we see here, over time follows the ICH M3R2 guidance. This is a guidance that you may have to read if you're called in to support the design of a non-clinical toxicology study. There is that guidance available how to design those animal studies. So these toxicokinetic studies are done in order to support a proof of concept study in humans. So the objectives, again, for developing a PD biomarker analysis is the effort of developing assay and measuring one or more markers across several phases is we want to validate target engagement, select dose, identify early efficacy, and identify potential subgroup who might respond and help design combination therapies. So let's look at PD biomarkers toxicology. So here we are often called to calculate the number of animals in various design, and I covered that. So there's serial design where serial sacrifice design where there's only one measurement available from the animal for one time point. And batch design is more prevalent, which is there's some time points available from some animals. Other animals are measured in other time points. So there is basically, the batch sampling is a sparse sampling. So not the entire time course is available. So we need special AUC and standard error of the AUC calculation. And Holder et al. Introduced a partial AUC method and a standard error of that. And also we need to, so basically the R code that I give in the book, this picture, for example, is plotting AUC hat over dose, and these AUCs and SEs are calculated using that R code using the partial AUC method. And so the data is read in and the code is given in the book to estimate the AUC, the SE, and the standard confidence interval. I also, so I go over the Holder et al. Calculation. I also introduce an R package PK, which does it with very, so I it does it with very simple calls. You can just choose which design, serial, complete, or batch, and it'll produce plots and the AUCs and the confidence intervals for you. So there are two ways I produce this. One is to actually code the Holder et al. Formulae that are asymptotically derived in the theory is derived in his paper. And also I use the same data set and use the function call to PK to give you the code to do it simply by using the R package PK. And PK actually is a very good package. It does all the non-compartmental PK analyses. And so I recommend that if you're involved in the early phase of toxicology and human clinical trial. The next, I go into the bioequivalence studies. So bioequivalents are also come up quite often to the biomarker biostatistician or to the early statistician to support. And there's several designs there where the designs could be parallel, multi-arm, replicate, or crossover. And the crossover design is very common, where the PK parameters are analyzed, and this TOST or TOST procedure, two one-sided tests. You can see here that the ratio of the mean parameters like AUC has to be within a certain bound, like 80% and 125%. And so this crossover study design and these hypotheses have to be conducted using R. These hypothesis tests have to be conducted. And this study is often called the 2x2x2 study, which I cover two treatments, reference and treatment, two sequences. Crossover means you can be reference, or treatment to reference, and two periods, your period 1, period 2 for every animal or person. So basically, I'm using the package Elmer test and Elmer, which is linear mixed models for this. I also give so the idea is that we have to compute the confidence intervals so that the FDA recommendation to stay within 80% and 125% is met for the two formulations of a drug, for example, for bioequivalence. And we have to compute the 90% confidence interval. And Schultz et al. In a paper shows with various examples that R and SAS and Win non-lin produce pretty much very, very close, if not exact output. Our package Power Toast provides power calculations, and our package BE will not only analyze the data, but will do the MSE calculation. So MSE calculations are also going to be important. That comes out automatically with that package, and I show how to do that. And as well as certain box plots of AUC. For example, here we have by sequence, by period, and by treatment. So both visualization and testing in this framework for bioequivalence is fully covered with one or two R packages that can be carried out. So I give the examples in the code in the book to be able to do that. Next, let's move on to more of the PD biomarkers. So the PD biomarkers is here in this example, you can see I have, let's say, 5 treatment levels. And I have a PD biomarker here. What is the exact endpoint? It's percent change from baseline at time equals T, like say 1 month after the treatment. And so this percent change is represented, and we can see that treatment level 3 and 5 has the biggest drop. Um, so 3 and 5 could be a potential appropriate, could be 30 mgs, 50 mgs that's going to go into the next clinical trial. So the visualization of this data, here I have give the code to do not only the mean and the mean AUC, the mean of the percent change from baseline, but I also plot the actual data. So it's in the same dose group. So this kind of visualization code is shown in the book, because I think a lot of biomarker statistician duty is to be able to let the data speak to the translational scientist and to the clinical scientist. So we have to spend time in making proper models, as well as the visualization, because it's going to be more than just calculating a p-value for our role in supporting drug development. So so now as we do this sort of cross sectional, when I say cross sectional, I mean at a specific time. So maybe 1 month is important in certain drug category, like insomnia. It could be 1 year in some other disease specific context. So basically, what I wanted to say is that if this endpoint could be anything. Here I'm showing percent change, but it could be AUC of that biomarker from time 0 to time t, or it could be the actual value of the biomarker, not the percent change, or it just could be the absolute change. So this is just an example of how to plot, how to make it good plot. And so sometimes we also we have to pay attention to comparing the means as well, because there's going to be multiple comparisons, right? So we could use something like Kruskal-Wallis, and to test whether the means and the medians are different by treatment group. And I use the R packet ggplot here. There's also a lot of newer type of displays are becoming popular, like violin plots and box plots. So those codes are given, and sometimes violin and box plot together. Like here, I have the mean and the actual data together. So you could play with that, the code I've given, so that the data is properly displayed. So now let's talk about longitudinal assessment. PD biomarkers are often collected over time. So we want to see the full time course profile of those longitudinal trajectories of each of those biomarkers. And so in the longitudinal analyses, I take really about 3 approaches. One, obviously, is the mixed effect repeated measures model, right? All of you are familiar with that. That's the approach 1. It's also abbreviated and called MMRM sometimes. And so I give the code to how to do that in R, how to plot the mean and the CI of the biomarker, whatever metric that might be. Endpoint could be the gain, and AUC, it could be percent change, it could be change, or it could be absolute value. But if the same biomarker is being measured in a particular subject, you do have to use the mixed effects model to account for the correlation within subject over time. Approach 2 is actually using the time varying covariate. Now, this was it's good for an exogenous variable, and it's been very well implemented in the time to event paradigm in the hazard modeling. So it's interesting because time varying covariate, it compares the risk of the event at each event time t between covariate set and risk set at time t. So it's looking at the covariate and the risk set at time t. So the covariate, which is in our case a biomarker, is allowed to um, vary over time and influence the endpoint. And therefore, that is a more powerful way of modeling a biomarker with the clinical endpoint. So that's something that I give code and explain in detail how that is important. And now the third example is the joint modeling of outcome and biomarker over time. And by the way, in the approach two, which is the time varying biomarker, that has been implemented by the Terry Thurno's R package survival. It's very, very easy to implement, but the math has been worked out by Terry using the partial likelihood mechanism. Yes, partial likelihood mechanism makes it very, very easy to implement. So for example, let me see here. No, I don't have it, but in the book there is an example where I showed that there is a 72.46% increase in the expected hazard relative to a 1 point increase in creatinine, keeping age constant. So in that model, if you use the time varying covariate model for your biomarker that is varying over time, you can also add uh, covariates that are not varying over time. For example, treatment, or even age. So or ethnicity, anything that is important to your model. So you can accommodate covariates that are both static over time and varying over time, and your outcome variable is your true endpoint of the clinical trial. So the joint modeling framework is to actually explicitly correlate the biomarker with the outcome. And so there's several papers out there, and I've implemented in the book the methodology laid out by Rizopoulos in the R package JM joint models, and applied to an AIDS data set. And we're going to talk about this more. In the AIDS example is very, very famous because, as you know, CD4 cell count had been correlated with as a biomarker, had been correlated with time to death from AIDS. So there was a joint model developed and for both of these, biomarker and the true endpoint. And I go through that example in R in the book. So this is just giving you a couple of plots to show you. So for example, this is the MMRM plot. You have a mean percentage change from baseline and over time. And this is using, again, mixed effects repeated measures, and using R to model, make a cool visualization. There are two treatment groups. And if red is placebo, we can see clearly that the treatment has driven down the value of the biomarker over time. This is a forest plot that I give code to do. So you can see here a hazard ratio of the onset of dementia. This data set is given and the code is given. Let's see what this shows. This is the hazard ratio forest plot. So this is the first row here is the biomarker percent change is positive. So biomarker actually increased, decreased 0 to -10, -10 to -20, and -20, greater than -20. So dipped below 20% from baseline. And you can see how the hazard ratio of the event of interest is varying. So for those who had biomarkers increasing at time t, so you can see that the first row, these is going to be mostly the placebo group. And you can, so some of this visualization can really be a powerful way of seeing how the biomarker is changing as in conjunction with the clinical endpoint. Yeah, so that is really the goal of a lot of this analysis for the statistician, is not just to um, make some plots and models, but to use the right tools to visualize and model in support of a story. And the story is um, being built with the scientist of how these biomarkers are working or not working in concert in the disease mechanism and also in the treatment paradigm. So I do talk about the multiple biomarkers as well in PD, in the section, or in the part of PD biomarkers. And I talk about the forest plots for multiple biomarkers. You'll find an example there. I do talk about network analysis, which I think is a novel way that I have found that is, I think, underutilized in clinical trials. Basically, these are Gaussian graphical models, and they're very, very good to understand the interrelationship between biomarkers. Often we say, hey, this biomarker is going to go up over time, this one's going to go down. Well, exactly how to do all of this work, and the network model of correlation between markers. uh, so how is, for example, percent change in BMI related to a biomarker one, sorry, percent change in biomarker one related to percent change in biomarker 2 and biomarker 4 and 5? Do they cluster together? Which biomarkers are related more closely because they're functionally close, or they're close in the pathway? Comes up automatically. This information comes out automatically if you use a network model of correlation or partial correlation. So I give the code an example for using network analyses for multiple biomarkers right from the PD biomarker part. And this is a topic that will come up again when we talk about the last part, which is combining biomarkers. And lastly, I use heat maps, which is a well-worn, used methodology in bioinformatics that can also be used to see how biomarkers follow each other. Yes, but I have found the network analysis to be more useful. With that said, let's move on to the second part. We have 4 or 5 parts to cover. So let's move on to predictive biomarkers. Now, whole books are written about predictive biomarkers. So I only scratched the surface of this. Predictive biomarkers, another word that is used often is called precision medicine. Precision medicine. So if the treatment is effective only for a subgroup, then it is our job as biomarker statisticians to find that subgroup and to tally the evidence that it's working in this subgroup and not the others. And when we establish that evidence over several phases of clinical trials, then it actually becomes part of the regulatory submission process. And it's a big deal, and it's a very complex it adds to the complexity of drug development. But the benefit of that is you might have this subgroup where the treatment works like gangbusters. So, there is also benefit to the complexity. So often we are involved in identifying the subset of patients based on a biomarker. We may know that there is this biomarker that measures, and maybe the higher values of these biomarkers are the patients that are going to respond to the treatment, but there may be uncertainty on how the lower values of the biomarker, those subjects would respond to that. So we have to identify a cut point or a threshold between positive versus negative. And we have to, and so we have to identify the cut point or the threshold. And we also have to do sample size calculations for either proof of concept or confirmatory trials with a biomarker that has positive and negative subgroups. There's also, I talk about one adaptive design. There's many kinds of adaptive designs where there's a seamless trial between stage 1 and stage 2. You can think of stage 1 as like a phase 2 study and stage 2 as a phase 3 study. In the stage 1, you are assessing the treatment effect in both positive and negative. And in stage 2, you will confirm that effect in the group, that either in the overall group or in the positive group. So those are some of the topics that I cover in that part. And this is probably by far the biggest part of the book. And so one question that comes up is how is it different from a prognostic biomarker? So prognostic biomarkers is like, right now I'm building a disease progression model. It's a multivariate model. But at the end of that, I have fast progressors, medium progressors, and slow progressors of disease. So that's a prognostic signature. It's different from predictive. Predictive is talking about who is going to respond, who amongst the disease will respond or not respond to the drug. And predictive biomarkers will also need drug diagnostic co-development. In other words, there has to be an assay, a validated test that measures that biomarker and produces reliable result as to who is positive and who is negative. So often that test has to be developed in parallel. Here's the drug development continuum, phase 1, phase 2, phase 3, and in parallel, the diagnostic test for the predictive biomarker also has to be developed if it's not already available. So let me just, I won't, so in the book, I don't go too much into the designs. There's a lot written about designing these trials, but most of the designs that are not adaptive fall into one of these three basic design. One is the biomarker identified group. So essentially you test by whether the subject is biomarker positive or negative. That means you already know how to identify this positive and negative. You have the threshold. Once you know the negative, then you don't enroll them. They're not eligible to receive the treatment. And in the biomarker positive group, you randomize to treatment and control. So one example is Herceptin in HER2 positive metastatic breast cancer. Another one historic is vemurafenib for BRAF V600E mutation in melanoma. These were the two first clinical trials that simply enrolled biomarker positive patients because there was enough science known already that the treatment would not work in the biomarker negative subgroup. Then there's a biomarker stratified design, and I call it it's also known as the interaction design, which is you look at you randomize both positive and negative. Again, you know who is positive, you know who's negative, so you know how to call, like if this biomarker is greater than this threshold, you're positive. If not, you're negative. So all that is known, the assay is available. You randomize, you stratify and you randomize both groups to treatment and control. There's many flavors of this design, but is the basic design. And here you are going to see either it's going to work in biomarker negative or it won't. And so after this trial, you can do a confirmatory trial. And then the adaptive design, where you test the biomarker in stage 1. If successful, go to stage 2. If not successful, stop. And so it's kind of like a ongoing trial. And we don't know when we start out which group is going to go to stage 2. So so now let's talk about some of the many issues that I cover in this part. First of all, the first bullet point, as you see, it is the operational characteristics of proof of concept trials with biomarker positive and biomarker negative subgroups is given in the book. And I use the R package, RPACT. And RPACT is actually stand for Design and Analyses of Confirmatory and Adaptive Clinical Trials with Continuous Binary and Survival Endpoints according to the methods described in the monograph by Wasmer and Branneth. It's a validated R package, so it will give you the same result as any other software out there that is also validated. So I designed in the example a phase 2 trial based on assumptions on biomarker positive, biomarker negative, and the overall population. So you need to make some assumptions. For example, hazard ratio maybe 0.9, in the biomarker negative group and 0.4 in biomarker positive group. So the overall, depending on the prevalence of the marker, maybe something like 0.7, 0.8. You can also model recruitment frequency, dropout, and so all those operational characteristics are obtained, which I provide. And here the power curves for above I generate so the power curves are going to be very important, right? Because we need to know, or your boss wants to know, how many subjects to recruit. And you also have to find out how many marker positive subjects you need to recruit. So I give the example for survival, but the same code and same package can be used for other endpoints. And again, there's a multiple. So the other thing is there's multiplicity involved. So the multiplicity, what is the multiplicity? The multiplicity is that you're going to test the biomarker positive and the overall group. Yeah, that's the multiplicity. There are many ways to handle that. One is the fixed sequence testing. If you do the fixed sequence testing, then that's fine. If you can agree on, so you almost have sort of an a priori idea of whether you want to test your positive group first or overall, or maybe the negative. And there's Bonferroni, of course, it's quite conservative. And there's Hochberg and Hom, which are better because it uses the rank of the p-value rather than a equal division of the alpha. But I give the R code for a lot of these, but I really get into a procedure for Dunnett test. And Dunnett procedure is something that I think is very powerful because it allows you again to take into account the correlation between the biomarker positive and the overall group, right? Because the biomarker subjects are also part of the overall population. So Um, Susan Halabi in her book gives an example of an oncology example, which I have found the data for, and I implemented the R code to come up with the exact same numbers. The correlation is between the test statistics of the subgroups that you're interested in testing. So you can see here on the right is the Dunnett test using the parametric Dunnett test. And there's two curves here. These are power curves. The red curve is for the biomarker positive group, and the black is for overall. So you can see that in 80% power is not reached in this example for the overall group, but it is for the biomarker positive group. So that is something that you may want to live with in a phase 2 trial. You can say, hey, look, we can only get 200 subjects and 140. We will have 80% power for 80% power for the marker subgroup, which everybody has agreed has the best chance. They think that the drug is going to work on that. So this code and the examples are given to give you sort of a flavor of the kinds of analyses you will spend a lot of time in if you're working in a predictive biomarker setting in phase 2 design. So that's one thing. Let's move on and talk about now something that I used to concern myself with a lot, which is if you do know the biomarker positive and biomarker negative group, maybe it's a mutation absent, present, or it's already well known. But sometimes you are in the situation where it's a continuous biomarker and you don't know where to draw the line, and you as a statistician is called in to start gathering evidence and on where to dichotomize that continuous biomarker. There's most of the statistical literature will say that um, you lose power. It's not a good idea to dichotomize a continuous biomarker. But lo and behold, that's exactly what we have to do. Often we're going to have an assay, we're going to screen patients, and we're going to have to declare potentially at some point that the drug is going to be beneficial on those subjects whose biomarker values is greater than, threshold. And it is not clear that the treatment effect just goes up with, if the treatment effect just simply went up with the biomarker value, then maybe it's not a good idea to do a dichotomy. But you may have an S-shaped where it is sort of latent or treatment effects very low, but at some point it starts to rise, and you need to find a statistically valid way for finding that cut point, for example. And in this case, if you have a continuous outcome variable, Y, you're really going to be interested in looking in this treatment by biomarker interaction term. And if both of these, the treatment, the interaction and the biomarker slopes are zero, the marker is no good. If the interaction is zero, then the marker is prognostic. So it's just a disease marker. It's not necessarily predictive treatment to predict marker. If the and you're interested in the last two situations essentially. And it's a very, very difficult job because often you don't have enough subjects to have enough power to reject this interaction term. So anyway, the point is to discuss how are you going to split the biomarker. And so there's the median. You can split it at the median, the quartile, the optimal split, and the step. There's many procedures. There's a rank conditional approach, and there is an interesting approach. So I discuss them. I also talk about the Holmgren procedure. And before, so I do look, usually you want to look at the interior between 20th and 80th percentile of the biomarker. Don't want to be in the extremes, and see where you're going to split it. I've given code for the bootstrap method, R package RCC, that gets not only the threshold where the treatment effect is the highest, but also confidence interval around the threshold. I also introduced a Bayesian method and a package called BHM to do the same. And just for this presentation, this is some of the different shapes of interaction that you can have. But for this presentation, I want to talk a little bit about the Holmgren method. Eric Holmgren is a colleague of mine from my old days at Genentech, and he came up with a very cool design, which I implemented in the book using simple R. It uses group sequential stopping boundaries to provide strong control of type 1 error. And how is group sequential method in interesting in this scenario? The way it's interesting is in interim analyses and in final analyses, where you use group sequential methods, you line up the subjects based on when they appeared in the clinical trial. So Eric used that same method, and he lined up the subjects based on their biomarker values. So let's say these are your hazard ratios, and lambda 1 is your treatment effect for all the subjects that have greater than that are in the highest greater than 75th percentile or above in the biomarker range. Lambda 2 is the hazard ratio for subjects who have biomarker values between 50th and 75th percentile, and so on. When you do that, actually, you get the four families of hypothesis testing: H1, H2, H3, and H4. And they have several components, as you can see. And you can actually use group sequential method boundaries and p-values and z-statistic thresholds to find out where in the biomarker region, at least which quartile, you have the highest treatment effect. So basically, the power and sample size and z-score thresholds are available, and I've implemented that in R, just using group sequential. You have a flexible alpha spending, and you identify the region where the treatment effect is the greatest. Once you find out, then you can design your phase 3 in the right zone of the biomarker as positive and the rest as negative. So I want to do a time check with Rick. Rick, can you give me an idea of how much time I have left?
Oh, sure. We still have another 30 minutes.
Oh, okay, great. So let's segue into the surrogate endpoint. Again, each one of this is many books have been written on this, and essentially we want to surrogate endpoints were biomarkers at one point, and NIH describe it as a biomarker that's intended to substitute for a clinical. Why would you want to substitute? Well, I mean, surrogate endpoints are used when the clinical endpoint might take a very long time to study, right? And also so that's one main reason it's used. And between 2010 and 2012, FDA approved 45% of new drugs on the basis of a surrogate endpoint, which is amazing. So the regulatory hurdle for getting a biomarker qualified as a surrogate is huge. So I don't want to underestimate that. I just want to introduce some basic analyses. And guess what? Even if you're not interested in establishing a biomarker as a surrogate endpoint, some of the analysis is very useful to see if one or more of these biomarkers are very close to the endpoint. Remember, we want to find out a lot about those biomarkers, and we're not always trying to get it regulatory acceptance of them, as sometimes we're just trying to understand the disease and the treatment process. There are two frameworks: the meta-analytic, which means it takes several trials and look at them across the trials to see if, for example, progression-free survival correlates with overall survival. The other one is within trial. So within trial, you have subject level data, and you want to see if the two endpoints, or the endpoint and the biomarker, are correlated. There's a third concept, is proportion of treatment effect explained. So if the treatment effect on the outcome and the treatment effect on the surrogate, what proportion of the outcome, a true endpoint treatment effect, is the surrogate endpoint treatment effect explaining? The best is 100%. The worst is 0%. Most biomarkers somewhere in between. So I talk a lot about that. So the FDA categorizes surrogate endpoints in roughly 3 hierarchical categories. You can have a candidate, you can have a reasonable likely surrogate endpoint, and you can get an approval based on that conditionally, or you have a validated surrogate endpoint, regular approval. A few examples of regular approvals based on previous surrogate endpoints: time to progression, progression-free survival, response rate. The other one I mentioned from AIDS was CD4 count. There's many. So let's talk about why we make such a big deal out of this. So you can see here that there are many situations. In scenario 1 is the ideal scenario. You have a disease, you've got the treatment coming in. Surrogate endpoint is in the pathway towards clinical. So it modulates and you capture it. In scenario B, the biomarker, the surrogate endpoint is not in the disease pathway, right? In scenario C, the intervention does impact the surrogate endpoint and then the outcome, but there's multiple pathways of the disease that modulates the clinical endpoint, and so on. So I won't go through all of them in the interest of time. The point is, it is not, what is the point? The point is, it's not enough to show that the biomarker is correlated with the endpoint of the study. Because in a lot of these situations that I show here, they will be correlated. That doesn't make it surrogate endpoint. So the Prentice criteria is really is a very, so you have to, if Z is the treatment and S is the surrogate, true endpoint is T, then you have to show that the treatment has an effect on the surrogate, treatment has an effect on the true, the surrogate has an impact on true, meaning all of these are correlated. And then this last one is very stringent. It says you're in a situation, because this says it completely explains the surrogate completely explains the treatment effect on the true, which was like our condition A, the picture A. You may be in a situation the surrogate only partially explains the treatment effect. So this is the same ideas, that I put in terms of a normally distributed surrogate has treatment impact. The true endpoint has a treatment impact. The true endpoint and the surrogate is associated. And then this true endpoint has now both treatment and surrogate. So this treatment effect, beta S, is what we're going to talk about. Yeah, here it is. Friedman proposed first that the ratio of beta S to beta, 1 minus that, is the proportion of treatment explained. So in other words, if beta S is 0, which is the ideal situation, your surrogate completely explains your true endpoint. then this is going to be 0, and then you'll be left with PTE is 1. So it's 100% explains. We're never really at that stringent criteria. So, so quantifying a biomarker between it requires two requirements. One is the trial level. You need to have data from several trials. Sometimes companies band together to get a certain biomarker established as a surrogate. So all the trials are looked at the same time. And then I give the example, our code an example, and then at the individual level, you have to assess the correlation as well. So in the meta-analytic, regression, you can show here R squared of trial equals 1, for example. It's never 1, right? You have different trials, and you have x-axis going in, for example, log of PFS, and the y-axis is log of OS. It's not going to all lie. The different trials are not going to be on your 45-degree line. But I go into how that this R squared, if it's less than 0.33, it's considered weak evidence. 0.33 and 0.67 is moderate, and above 0.67, strong. And here in the R squared individual, Kendall's tau is used. 0.61, for example, is considered, strong. And I do introduce something called a copula model with the Clayton copula, where you can model again the surrogate biomarker and the true endpoint as a bivariate random variable. And so therefore you can make use of the correlation and come up with a good estimate of the R squared individual. So so basically for example, the S and T here is considered as a paired variable, and it's a two-stage approach. Yes, it's a two-stage approach, because you model the for example the hazard based on the true endpoint as modulated by Z, the treatment, and the second is the hazard modulated for the surrogate by treatment. And the third is you specify a function, how the copula function, where the H of S and H of T for true are tied together. And this is implemented in the code. And I give an example, and this is so Michael Hughes, and in his seminal paper, introduced the two methods. You can actually determine proportion of treatment effect explained on the AIDS clinical trial. He has his true endpoint and CD4 cell count. And Beth Zamboni, in her PhD dissertation, revealed a nifty trick that you can actually use the regular hazards model to look at the paired variable S and T and get estimates for beta S and beta. Now, the way that Michael Hughes estimated PTE and CI is using a delta method, and I give that code. But also, I've introduced a new method called in Bayesian linear regression, which gets a very similar, which fits the same models, the two-step approach, and also gets a very similar PTE and confidence interval estimated. So I think you will find these codes very useful. Here is a example from book where I've given the code to not only derive this, but also plot the difference in mean change in CD4 at 6 months and the hazard ratio. So the higher the change, the lower the hazard ratio. And so this is the Bayesian approach. So each of these are different trials. And so we actually find out the intercept does not include zero. So the intercept actually should be zero. And the slope is non-zero. So, and that's how we want that. And so these are some of the statistical methods underlying the analyses of a biomarker that you and your scientists, you as a data scientists and other scientists have want to show as not perfect correlation, but very, very close to the disease pathway that is being impacted by the treatment. Let me move on to the combination of biomarkers. That is, again, many books are written in machine learning. I do go into, a few models that I keep using in the context of clinical trial. What is machine learning? Field of study that gives computers the ability to learn without being explicitly programmed. This is where you may find some of the more traditional biostatistical principles don't apply here. And so you might have to do a lot of education and also education of yourselves. So the idea is that these models will tell you, for example, which biomarkers it considers to be important, playing a role, for example, in treatment effect on the true endpoint. So you have to let the algorithms find the right predictors and the right models, the function, how to get the biomarkers, combined in a model that predicts the outcome. Could be the treatment outcome, it could be the disease outcome, in which case you are building a prognostic signature. You could be building a predictive signature or a prognostic signature. You could be making a diagnostic signature where you want to, for example, we're building a model where we're predicting who's going to be amyloid positive, amyloid negative. This is going to be used in the screening phase. So I introduced regularized machine learning. And I first start out with, let me just see here. Yeah, I first start out, okay, I think I've talked about regularized machine learning and then unsupervised and then graphical models. Remember, I told you I'd come back to graphical models. So let's talk first about regression-based methods. So the regression-based, I start out with best subset selection, which most of you know, forward, backward, best subset. Also introduce the modern ways of cross-validation. I give a synthetic data set where I'm trying to find a safety signal. So I'm in the world of logistic regression. You can use best GLM to find the best subset, given that your predictors are not very many. And that is usually the case for biomarker statisticians. We're not in the world of bioinformatics usually. So there's family-wise error rate correction that and I also use the 1SD rule to find out what is the size of the best subset. So if you have 10 biomarkers, how many do I pick? Maybe 3, 4, depending. I don't pick it. The tools I introduce tell me what is the size of the subset and who are the important biomarkers. Do introduce the ridge, lasso, and elastic net as the regularization and talk about how to determine the tuning parameters. And so I do a lot of this by hand, and later I introduce R package, Carrot, and there are many other R packages that but you know that Carrot is very good because it has very nice hooks or functions to do the tuning and the cross-validation in a coherent and consistent manner. So I do want to say, though, it gets a little bit complicated because we are in a world where we might want to detect interactions, treatment by biomarker interaction, which biomarkers are being modulated by treatment, especially in the predictive models. So I talk about hierarchical group lasso, and I introduce this package GLM. Actually, it is GLM interaction, I believe, to find the right interaction term. Because we're in the regression-based context, we are not just interested in the main effects of the biomarker, but also in the interaction. Let's move on, talk about tree-based methods. This is really good when you want to do the non-parametric ways. And these are usually greedy algorithms, so it's a divide and conquer strategy. Um, I use first the R package rpart, which is very rudimentary and basic, and it prevents overfitting from minimal cost complexity pruning. And I build up to classification trees and hazard survival trees, and then the ensemble of trees like random forest. The cluster analyses I also introduced. These are unsupervised methods. So perhaps we're interested again in just looking at the interrelationships between the biomarker, maybe between two conditions, maybe just looking at the treatment group. How did the biomarkers change at 1 year or 6 months or 1 month? Unsupervised methods are extremely useful to cluster the biomarker data. And the algorithm can find underlying structure in the data, whether they're genes or biomarkers, without the label. So there are three methods. I talk about the k-means, the PAM, which is partition around midoids, and hierarchical clustering, which I find extremely useful, hierarchical clustering, but you do have to give it the number of clusters. And with the other two, k-means and PAM, you get to find out how many clusters there are. I talk about distance metrics and linkage methods, which are uh, two parameters you have to pay attention to when you are doing agglomeration of clusters in hierarchical clustering. Then again, back to the graphical method. So this is a relatively novel method. You can call it graphical method or network analyses. And I see that you can actually enter treatment and endpoint and these biomarkers. And let's say that the green edge here, these are acausal undirected graphs that I made through our package iGraph. And it shows that this biomarker is positive, that as you go higher in treatment can be ordinal, let's say, mgs or levels. um, 0 is placebo, you have 2 levels of treatment, 1 and 2. A biomarker is positively correlated with the treatment. And so this biomarker 1 and treatment, so it is the higher the treatment, lower your endpoint, and and also the biomarker. So so this thickness of this is the strength of the relationship. But what is the edge? The edge is a statistical quality, like quantity, like correlation or partial correlation, which is the one I prefer. And the nodes are variables, so random variables like treatment, or Y could be change in a score, change in disease at age 1. BM1 could be change in biomarker at year 1, and so on. So it detects the association between variables, and the color of the thickness is the strength of that partial correlation. Bigger it is, the thicker. And the color denotes the direction. And and you can find these triads or networks of biomarkers. You can find the topology of all the biomarkers. And it's been used in a lot of fields, but it's underutilized in clinical trials. As we get more and more biomarkers, we should be using graphical methods to elicit the interrelationships in this way. Now, genetics researchers have been using it, and You can also regularize this network, which is a beautiful thing by itself. So graphical lasso by Tip Shirani is available to penalize this graph for doing multiple and cyclical partial correlation calculations. So a biomarker analysis plan is something that I really recommend that you develop. You should be the person um, developing this, and again, in collaboration with your colleagues. You will develop this with your translational science team, clinical science team. The scientific rationale for each biomarker should be included. If it is being measured and paid for and the data is coming in, there has to be a scientific rationale. So this would be a good place um, to capture that. And it should have a minimal overlap with the study protocol and the main SAP. And again, some of the mains, for example, if you have a predictive biomarker, biomarker positive, biomarker negative, that analysis may very well be captured in the clinical SAP, but all the other analyses can be captured in the BSAP. So there are 7 sections that I took from, that I borrowed from the clinical SAP. So there should be, I recommended you can, delete and add sections as you need, but introduction, study design with an eye towards anything related to biomarker rationale. Fourth is objectives. Fifth is endpoints. So for example, what endpoints is it going to be percent change at 1 year? Is it going to be AUC from 0 to 1 year? Things like that. Endpoints, general considerations, how you're going to impute missing values, what kind of normalization, if any, you will do. For SNP, you might have SNP data, you might have IHC data, immunohistochemistry data, you might have blood-based biomarker. Are you going to do any pre-processing? And then the last section would be statistical method. So if you are developing a diagnostic signature or a prognostic model, if you're going to do MMRM plots, if you're going to do graphical models, so the objectives section should be tied to the statistical methods. And there's 7 sections that I proposed that I go into that. And then last but not the least, misuse and abuse of biomarkers, you should be aware of. Mostly I see them as incomplete use. So the totality of the evidence of biomarker collected is often ignored in the clinical trial. We need to look at that, and we could do that by having a systematic plan ahead of time for biomarker analyses and collection. Wrong use. So for example, target engagement does not mean efficacy. And I hope that's clear from this talk. We're measuring different things at different times with potentially different biomarkers. So and then also a surrogate, something that is correlated. For example, a biomarker that's very well correlated with the endpoint does not mean it's ready to be surrogate. As you saw, there are many steps for that. There's also a problem of lack of use. So there, you could be, as a statistician, seeing that there is a group, but that where the treatment is effective, but for some reason, the company is not embracing the predictive group, and they may be ignoring a precision medicine approach. But that's really where the industry is going in oncology, immuno-oncology, in cardiology. If the drug is working in a specific group, then it's worth looking at instead of that broad claim and having sort of a less than mediocre treatment effect. The other, I wouldn't call it misuse or abuse, but I would say that we also need to keep educating ourselves as new modalities of biomarkers are coming in the picture. especially digital biomarkers and imaging. So we need to be part of a culture that is continually educating ourselves as the instruments are getting higher and higher precision, as the disease biology is getting more and more advanced, and so should the methods to keep up with these a plethora of information that's arriving from clinical trials. So with that, I will stop. I do want to show you that I've given you the book reference here, and I have a podcast that is already available, episode 79. And again, thank you for the attending the webinar. And I want to acknowledge all my industry colleagues over the years that I have, collaborated with, as well as I wouldn't be able to do what I do without the world of R and the amazing number of papers on stat methodologies that exist. And all the academic folks that are churning out methodologies and implement computational methodologies, implementation of those methodologies. So Rick, should we listen to some questions?
Yeah, let's do that. So a couple have come in. The one that was earlier, I think you may have covered already, but I see a couple that have just come in recently if you want to take a look at those.
So, so how would one handle longitudinal PD biomarkers collected at different time points and each containing measurement at different days after therapy? Would time varying covariates be the best way? So yes, I think that time varying covariate, which again is implemented um, in the time to event methodology by Terry Thornow's package Survival in R. Also, if they're collected at different time, everybody's at a different time. You can also consider bucketing them. So, for example, biomarkers collected in the first 3 weeks, or making a coarser bucket instead of looking at every measurement, which is different for a different visit, even is what you're saying for different patient. You might say, okay, let me look at the mean of the measurements in the first 3 weeks. So you have to get creative, because in the models we are, in the business, sort of in predicting the average effect. So, and it's kind of hard to do that if everybody's trajectories are all over the place. So you can also define endpoints as the increase up to and including 1 year. So it may not be possible for you to do longitudinal trajectory unless you want to plot it for every patient, and unless you come up with some ways of standardizing or stabilizing the different time points for the markers. How large, this is a great question, how large do studies need to be to use network analysis? Can it be used for small exploratory studies? From Kim? Actually, yes. Kim, I'm glad you asked that question. It is actually very good for small and exploratory studies. I've used it because it's very, very powerful. That's the good point of it. The benefit of it. And so the issue is that when you regularize, because you need to regularize, right? Because you're looking at multiple edges between variables. In the regularization parameter for graphical lasso, you make sure not to over penalize when you have a small number of subjects, because you over penalize, it'll just show no edges at all. So you have to kind of play with that, and have dial it up just enough. So I'm not saying that doing the network analysis will get you the exact topology, but you want to get the primary edges out of that analysis. And if there's only one tuning parameter from what I know, if you tune it just enough, you should be able to see the important edges. Carlos, do you think it's time to change the paradigm, make the biomarker the clinical endpoint and the clinical outcome the surrogate endpoint, especially with the advent of machine learning? That's a really great question, Carlos. I think especially in the COVID-19 vaccines and other, drugs, COVID-19 treatments, depending on the rare disease, depending on the context, in the rare disease and also emergency use context, the FDA has been very open to approving conditionally on biomarkers. So then it's up to us to really then later on go and establish the relationship with the true endpoint at the end. And another thing you say, it's a very interesting question, because when you have many biomarkers, it may even give you more information about what's happening in the body than the clinical endpoint. But again, it's a high hurdle to change the regulatory signs and have them accept many biomarker evidence. It's only now that we are collecting these blood biomarkers and other fluid biomarkers. One of the reasons is that the molecular diagnostics companies working on these tests and instruments have gotten so good that these information are being collected, and it is possible that at some point this gives us more information than our traditional endpoint. We're not there yet. Zin has asked a question, how to construct the network topology. Yes, so I think that in both in R and in Python, we can look for graphical Gaussian models or Gaussian graphical models, and look for building partial network analysis. It's very freely available in R. The package is iGraph. You feed the data in it. Actually, it's a machine learning approach. It gives you the network. Inayat is speaking and saying, thank you for this presentation. Is this book a good starting point for somebody? Yes, it is. It is a good starting point because it's not an exhaustive, I don't think it's possible you have to be an encyclopedia. It is a good starting point because it covers from toxicology all the way to combining biomarkers. So you're likely to find something useful there for your work if you use it. QE Shen, thanks for the talk. I have a question about FDA submission. Is R allowed for FDA submission? A lot of the R packages are validated now, so they are cleared for submission. And also, if you recode, if you use a package that is not validated, then you can actually use basic R to program it and give your program to the FDA for acceptance. So FDA is becoming more and more accepting of other software for submission. Anonymous, I had a quick question about calculating hazard ratios for endpoints. What is the best way to handle recurring endpoints longitudinally? Well, that is not a biomarker question, but let's just say that in Terry Thurno's package Survival, you not only have time varying covariate in hazards analyses, you also have traditional survival is, you have an event and that's it, or you become censored. But you can actually have endpoints that are recurring. Yes, so you can have competing events and recurring events in our package Survival. You have to turn on those modes, but you're completely able to model those phenomenon and find out if the treatment made any difference. So that is one of the big contributions of Terry Thurnow from University of Washington. Okay, I think, Rick, those were all the questions. And maybe it's a good time to stop for today.
I think so. I think it's a perfect time. Dr. Rabbee, thank you very much for the presentation today. We really appreciate it. And thank everyone for attending. I hope you'll be with us for more of these webinars as we schedule them. We have plenty coming up. So everyone, I'm going to shut down the webinar. Have a wonderful day. Goodbye now.
Thank you.